All right, today we're going to go over the study guide for the Unit 8A exam. So first we're going to go through some vocabulary. Number 1 says, this is the shape of a typical DNA molecule when it's in the nucleus of a eukaryotic cell. This is when it looks like a twisted ladder, and the name of that shape is E, the double helix. Number 2. This is the process that occurs when a DNA molecule makes a copy of itself with the help of enzymes. So when DNA is being copied, the name of that process is replication. So number 2 is F. Number 3. This molecule stores genetic information in cells and is stored in the nucleus of eukaryotes. So this is DNA, right? That's the name of that molecule, and so the letter answer is D. Number 4. This is a virus that infects bacteria cells. So those are referred to as bacteriophages, so the letter is A. Number 5. This is a process where DNA from one bacterial cell is taken up by another bacterial cell and expressed. An example is what happened during Griffith's experiment. So number 5 is B, and that is called transformation. And number 6 says this is the place in a eukaryotic cell that stores DNA. So in eukaryotes, like plant cells and animal cells, you're going to find DNA stored in letter C, which is the nucleus. Next, we're going to review Griffith's experiment. So we're going to go through several different DNA discovery experiments. We're going to start with Griffith. So if you recall, when Griffith did his experiment, he injected mice with 2 different types of bacteria. So one of the types of bacteria he used was a rough strain, which is also non-virulent. Remember that when something is non-virulent, it means that it does not cause disease. And then he also injected mice with a different bacteria strain called the smooth strain that was virulent. And the outcome for the mouse was different with each of those strains, right? What he discovered is that if he killed the smooth bacteria, the outcome was different than if he injected live bacteria. And he also discovered that if he mixed together the rough strain and the heat-killed strain of smooth bacteria, he had a different outcome. So we're going to review those outcomes now. Number 7 says write the letter or letters that would represent a mouse dying. So when a mouse was injected with the rough strain of bacteria, like he is here at letter A, the mouse lived, so we don't have to write that one down. When the mouse was injected with the smooth strain at letter B, the mouse died because smooth strain is virulent. So we know that B is one of the correct answers. And we also know that if he killed the smooth strain, it did not kill the mouse. So letter C, the mouse lives. And when he injected the mouse with the rough strain plus the heat-killed smooth strain, he discovered that the mouse died at letter D. So the mouse died in both cases, B and D. Letter 8, or number 8, says write the letters that would represent the mouse living. And as we just talked about, when the mouse was injected with the rough strain or the heat-killed smooth strain, the mouse lived. So the mouse survived at both A and C. Number 9 says what process occurred between the heat-killed smooth bacteria and the live rough bacteria in mouse D. So this is where we learn about that process called transformation. So this is where the DNA from the dead smooth strain entered the cells of the rough strain, which was still alive, and it caused the rough strain to express the genes from the smooth strain. And so the name of that process is transformation. And it's when DNA is transferred from one cell to another and then expressed in the live cell. For the next several questions, we're going to look at the diagram that is provided and answer the questions. So number 10 says, what is this? So that's referring to the image above, and that is a bacteriophage, which is a virus that infects bacteria cells. And so that also answers our number 11 question, what does it infect? So bacteriophages specifically infect bacteria cells. They don't infect human cells because we have eukaryotic cells. Number 12, which part of it contains the genetic material? So if you notice, you can see all the different structures labeled. We know that genetic material is DNA, so we just have to decide which part of the bacteriophage contains the DNA. And so since the DNA is enclosed inside of this structure here, the DNA is contained in the head of the bacteriophage. Number 13 says who used it in their experiment. So if you recall from the DNA scientists that we've learned about, there's two scientists named Hershey and Chase who were able to tag DNA and proteins with radioactive phosphorus and radioactive sulfur, and they used a bacteriophage with these radioactive labels to infect bacteria cells to find out which part of the virus went into the cell to cause an infection. So they were tracking where the DNA went by using radioactivity. And then number 14, because of their experiment, we want to know what molecule did Avery conclude causes transformation. And so what they discovered is that it is in fact DNA. So letter A is the answer. They discovered that DNA causes transformation. Next, we're going to look at DNA structure. So here we're going to use this diagram, and it's a specific molecule. So number 15 says, what molecule is this? And this is, in fact, a DNA molecule, right? It has that very characteristic twisted ladder structure. And number 16 says, where is it located in eukaryotes? So this is in the nucleus, right? So in eukaryotic cells, DNA is always contained in the nucleus, and it does not leave the nucleus unless a cell is dividing. Number 17 says, what pairs with guanine in the molecule above? So if you remember, there are 4 DNA bases that contain nitrogen. So there's adenine, thymine, guanine, and cytosine, and they pair up with each other in a very characteristic manner. So guanine always pairs with cytosine, and then adenine always pairs with thymine. Number 18 says the percentage or amount of adenine always equals the percentage or amount of thymine in the DNA molecule, right? And that's because adenine and thymine are always found across from each other on the 2 sides of the DNA structure. And so then that means that number 19, the percentage or amount of guanine, always equals the percentage of cytosine in a DNA molecule. On this page, we're going to answer questions 20 through 25 about DNA replication. So number 20 says what is shown, or what process is shown in the diagram above. And the name of that process is replication. So that's what it looks like when DNA is making a copy of itself. Number 21, what is the name of the enzyme that unwinds the DNA? So there is an enzyme that opens up the double helix to expose the bases, or the adenines, thymines, guanines, and cytosines on both strands of the DNA molecule. And when those bases are exposed, a new strand can be built. And the enzyme that is responsible for that process is called helicase. Number 22, what is the name of the enzyme that adds base pairs to the strand? So once the enzyme's helicase has opened up the DNA strand and exposed the bases on both strands, then a different enzyme comes along and adds bases to build a new strand based on the existing strand. And the name of that enzyme is called DNA polymerase. Number 23. What is the name of the enzyme that binds the Okazaki fragments from the lagging strand together? So remember that when DNA is being built, the 2 strands that are being built in opposite directions. And one of those strands is called the leading strand, and one of those strands is called the lagging strand. Remember, the lagging strand is the one that's growing away from the replication fork. And so the pieces that are being built from the lagging strand need to be tied together before you have a complete DNA strand. And so the enzyme that can tie those fragments together is called ligase. Number 24. What nucleotide will be added to the top strand next? And so the next base that is available here is the letter T. So if we have a T on the template strand, it means that we're going to add an A, or an adenine, to the new strand that's being built. And number 25 says write the DNA sequence that would be formed if you're given the following. So our template strand, the existing DNA strand we have to work with, has the bases CAT, GAC. So now we need to write the complementary base. So on the template strand, if you have a C, the new strand will have a G, right? Because it's the one that's across from the letter C. And then if you have an A, the next base will be a T. If you have a T, the next base will be an A. And then if you have a G, you will get a C on the new strand. A matches with T. And finally, a C will match with a G. So the new strand should read GTACTG. Number 26 through 28 are looking at the structure of a nucleotide. So if you remember, DNA is made of a series of nucleotides that are bonded together. And so we're just going to identify the parts of a nucleotide in these questions. So number 26 says which letter represents the sugar. So the sugar in our nucleotide is letter A. And the sugar has that pentagon shape. It's called a pentose sugar. This one happens to be a deoxyribose sugar, which is part of how DNA gets its name. And so that's the sugar that's in each nucleotide, that's each step of the ladder for a DNA molecule. Number 27, which letter represents the phosphate? So the other part of the backbone, essentially, of the DNA molecule is a phosphate, and that is letter B. And then number 28 says which letter represents the nitrogen base. So remember, the nitrogen bases can change. They can be adenine, guanine, thymine, or cytosine, but they're always in the same place on the molecule, and that is at letter C. Finally, we'll talk about how the DNA molecule is conserved as the DNA is replicated. So number 29 says which type of replication is used when DNA is copied in a eukaryotic cell. So it could be conservative, semi-conservative, or dispersive replication. And in eukaryotic cells, the reason or the way this happens is through semi-conservative reproduction or replication. And that's because when you start to make a new DNA strand, you take the original strand, which is called the template strand or the parent strand, and when that is replicated, the two strands split apart and they each build their own new strand. So you have one template strand that's represented here in blue in each parent, and then you also have a new strand that's represented here in yellow, and those two strands together make the new DNA strand. And so that's semi-conservative because some of the old DNA is used in the new molecule. And then number 30 says what percent of a new strand of DNA is composed of the template strand. And so if you look, you can see half is yellow, half is blue, meaning half is old and half is new. So that is 50%. So again, that's because DNA has a semi-conservative way of replicating.